at BSc, what we're working on is in the cancer use case. So we want to bridge from the proteins to the more organ level physics of cancer growing in the brain. We want to simulate from signaling pathways up to tissue levels. So we do this by building up different signaling pathways that are important in medulloblastoma and how these effects or how perturbations on these signaling pathways affect the cancer growing in the brain. And we want to couple that to physics of the cancer growing in terms of tissues, in terms of tensions, in terms of pressure of the cancer in kids' brains, let's say. So what we have been done is to personalize different models of medulloblastoma for different patient data. So that in order to have different kinds of patient-wide, I would say, simulations in which you have a family of models that characterize different aspects of the different patients, and how these different patients have different simulations in terms of how we can treat that patient, how we can intervene in the cancer growth, and how we can at the end inform medical users or medical clinicians in order to fight this cancer. One of the big obstacles that we're facing is that we're bridging lots of scales, lots of time and spatial scales. So we're working from protein functions up to the pressure of the cells growing in the brain. So that means that there are lots of assumptions that may be true in one part of the simulations and not in another part of the simulations. So in order to scale, to bridge those scales, we need to be very mindful of the different assumptions of the different levels. And in order to combine different variables from one scale to another. So we're working with collaborators in EDITH and in order to connect different models that are focusing on those different parts of the let's say overall model.